In this video, we will talk about the sex linkage. Traits controlled by alleles on the sex chromosomes are said to be sex-linked, and the phenomenon is called sex linkage. An allele that is only on the X chromosome is X-linked, and the allele that is only on the Y chromosome is Y-linked. Some traits are controlled by such genes which have alleles found on both X and Y. Such traits are called XY linked or pseudo-autosomal, because unlike other sex-linked traits, they have just like autosomal mode of inheritance. Very few alleles have been found on the Y chromosome. Now let's discuss sex linkage in Drosophila. Morgan's experiments on eye colour in Drosophila. Letter capital R represents the dominant allele for red eye, and letter small r represents the recessive white eye, because the alleles are carrier on the X chromosome. These are shown as superscript to the letter X. Thus, the red-eyed male fruit flies have genotype X capital RY. White-eyed males are X small ry. The Y chromosome does not have a gene locus for eye colours, therefore. The phenotype of the male results entirely from this single X-linked genes. In the female, X capital R, and X capital R, and X capital R, and X small r have red eyes, and X small r, and X small r. Flies have white eyes. First, Morgan crossed the white-eyed male to a red-eyed female. About 1237 flies were produced in F1 generation, and all were red-eyed. This side for female, and this side for male. So, all were red-eyed. Next, he crossed between F1 generation to produced F2 generation. This side is for female, and this side is for male. Both females are red-eyed, and one male is red-eyed, and one is white-eyed. Now, would the white eye trait reappear in the F2 progeny, as Mendel had predicted? In the F2, there were 3,470 red-eyed flies, and 782 white-eyed flies. In this experiment, he observed two deviations from what Mendel had explained. There was a slightly deviated phenotypic ratio in F2 generation from standard, 3:1, of monohybrid cross. Red-eyed are 3 times more than white eyes. And secondly, that all the white-eyed flies in F2 generation were male. Once he thought that perhaps the white-eyed trait somehow killed female flies preferentially. Morgan preferred a straightforward test. If any of the F2 females carried the white trait, did not show it, then it should be revealed by test cross to recessive parent. It was crossing red-eyed F2 females. Back to the original white-eyed male. He obtained 129 red-eyed females, 132 red-eyed males, and 88 white-eyed females, 86 white-eyed males. Again, this was a rather poor fit to the expected. 1:1:1:1, due to a deficiency in recessives. The important thing, however, was that there were fully 88 white-eyed female flies. This observation finally discarded his doubt that the white-eyed trait can kill female flies. Appearance of white-eyed female provided an opportunity for a further confirmatory test. Morgan mated a white-eyed female with a red-eyed male, all female offspring had red eyes, and all male offspring had white eyes. Morgan's concluded. A simple explanation of his results, Morgan concluded that the white eye trait gene resided on the X chromosome. Morgan had only to assume that the Y chromosome did not have this gene. Knowing from his previous crosses that white eye is a recessive trait, the results he obtained could be seen to be a natural consequence of Mendelian segregation. Now let's discuss sex linkage in humans. Humans have many X-linked traits, of which some, like hemophilia and color blindness, are recessive, while others, like hypophosphatemic or vitamin D-resistant rickets, are dominant. X-linked dominant is a trait which is determined by X-linked dominant gene, while X-linked recessive is a trait that is determined by X-linked recessive gene. Their patterns of inheritance are very different from each other. Experimental mating is not practically possible in humans. Mode of inheritance of human traits can be traced through pedigrees. Before moving to X-linked traits, let's discuss Y-linked inheritance. So, Y-linked traits are called holandric traits, the genes located on Y chromosomes, and are absent on X chromosome, called as Y-linked genes or holandric genes. The Y-linked genes are located only on the non-homologous region of Y chromosome. Their phenotypic expression is observed only in the males. They are entered from father to son, as son only receives Y chromosome from father. The examples of Y-linked characters in men are hypertrichosis, which is growth of hair on the rim of pinna, and webbing of toes. Let's have an example of hypertrichosis. This side is for female, and this side is for male. All females are normal, as both the X chromosomes from mother and father are normal, while all males are suffering from hypertrichosis, as only male child receives Y chromosome from father. Y chromosome has this trait of hypertrichosis. Now let's continue the sex-linked disorders in human. Human have several disorders which are caused by the mutation in sex chromosomes, as Drosophila has white eye color. The inheritance patterns of some sex-like disorders in human are genetics of hemophilia and genetics of color blindness. First is genetics of hemophilia. Hemophilia is a rare X-linked recessive trait. Hemophiliac's blood fails to clot properly after an injury, because it has either a reduction or malfunction or complete absence of blood clotting factors. It is a serious hereditary disease because a hemophiliac may bleed to death even from minor cuts. Hemophilia is of three types: A, B, and C. Hemophilia A and B are X-linked recessive, but hemophilia C is an autosomal recessive trait, being X-linked recessives. Hemophilia A and B affect men more than women, but hemophilia C affects both. Now let's discuss types, causes, and inheritance of hemophilia A, B, and C. Hemophilia A is caused by abnormality of blood clotting factor VIII, and its pattern of inheritance is recessive, X-linked. Second is hemophilia B. It is caused by disturbance in blood clotting factor IX, and its pattern of inheritance is recessive X-linked. Third is Hemophilia C. It is caused by reduction in blood clotting factor 11, and its pattern of inheritance is recessive autosomal. Now let's discuss treatment. So, cases of mild hemophilia A can be treated using a type of medication called desmopressin. Desmopressin is a synthetic hormone. Desmopressin works by stimulating the production of clotting agent 8 and is usually given by injection. Hemophilia B, which is caused by a lack of clotting agent 9, it is treated with a medication called nonacogalfan, which is an engineered version of clotting agent 9. Hemophilia A is treated by using a synthetic version of clotting agent 8 called octacogalfa, which is another type of genetically engineered purified protein. Let's have an example to understand its pattern of inheritance. Well, X chromosome with capital H is normal, and X chromosome with small h is hemophiliac. Female can be normal, or hemophiliac, or a normal carrier, but a male can be normal, or hemophiliac. So, what will be the result? If normal female marries a hemophiliac male, so this side is for female, and this side is for male. Both daughters will be normal carriers, and both males will be normal. So result is that 50% female normal carrier and 50% normal males. Now let's discuss the genetics of color blindness. Normal trichromatic color vision is based on 3 different kinds of cone in the retina, each sensitive to only one of the 3 primary colors: red, green, or blue. Each type of cone cell has specific light-absorbing proteins called opsins. The genes for red and green opsins are on X chromosome, while the gene for blue opsin is present on autosome 7. Mutations in opsin genes cause color blindness, like dichromacy and monochromacy. A dichromate can perceive 2 primary colors, but is unable to perceive the one whose opsin is missing due to mutation. Protanopia is red blindness. Deuteranopia is green blindness, while tritanopia is blue blindness. And a monochromate can perceive only one color. Monochromacy is true color blindness. Blue cone monochromacy is an X-linked recessive trait, in which both red and green cone cells are absent. That is why it is also called red-green color blindness. It is a common hereditary disease. This is normal vision. But this is vision affected with red-green color blindness. Like any sex-linked recessive trait, it also zigzags from maternal grandfather through a carrier daughter to a grandson. It never passes direct from father to son. This type of color blindness is more common in men than women, because chances for a male to be affected by it are double than a female. Now moving forward. So, which of the following pair of parents is most likely to produce a daughter with red-green color blindness? Carrier mother and unaffected father. Unaffected mother and colorblind father. Colorblind mother and unaffected father. Carrier mother and colorblind father. Let's understand it. Carrier mother and unaffected father results in carrier female, normal female, carrier male, and normal male. Next case is unaffected mother and colorblind father. Results in carrier female, normal male, and normal male. Next case is colorblind mother and unaffected father. Results in carrier female, carrier male, and carrier male. Next cases: carrier mother and colorblind father. Results in colorblind female, carrier male, and normal male. So, carrier mother and colorblind father. Most likely to produce a daughter with red-green color blindness. This was all about sex linkage. In our next video, we will talk about the sex-related traits.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.